Hello, everyone, and welcome to today's webinar, Applied Biosystems TaqPath COVID-19 Combo Kit, a highly sensitive, specific, and robust real-time PCR test. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher. To learn more, visit www.thermofisher.com. So let's get started. Before we begin, I'd like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you can. To do so, simply type them into the Ask a Question box and click Send. If you have any trouble seeing or hearing this presentation, click on the Support tab found at the top right of your presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I now present today's speakers. Lauren Mikulski, Senior Director in the Product Management Genetic Science Division, Clinical Solutions at Thermo Fisher Scientific, Dr. Vijay Singh, Managing Principal Scientist for Molecular Diagnostics at Healthtrack RX, and Dr. Andrew Brooks, Professor of Genetics, Rutgers University at RUCDR, Infinite Biologics, CSO Brooks Life Sciences. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Welcome to all of you. Lauren, you may now begin your presentation.
Good morning and good afternoon. My name is Lauren Mikulski. I'll be speaking today on the Applied Biosystems TaqPath COVID-19 Combo Kit. I'm excited to discuss the solution for detection of SARS-CoV-2, the virus that causes COVID-19, from patient samples using gold standard real-time PCR technology. When we started looking at this, when the coronavirus outbreak began in late 2019, Thermo Fisher Scientific rapidly developed one of the first COVID-19 diagnostic tests, which is now scaling to manufacture 20 million tests per week. Thermo Fisher has scaled with the COVID-19 global crisis and has been able to provide consumables, reagents, instruments, and software to customers around the world. Thermo Fisher is working to apply its scale as a world leader in serving science to standardize and industrialize the manufacture of tests and testing platforms in a reliable and reproducible way to support the global need for testing at scale. As you can see here on the slide, globally, over half of all COVID-19 testing is currently being performed on some of the most used real-time PCR instruments in the world. We have rapidly increased our production of COVID-19 kits and continue to increase our manufacturing as needed in preparation for the next respiratory season. As you've heard in the news or maybe experienced yourself, laboratories have been constrained with supplies from reagents, consumables, and instruments. Thermo Fisher provides the end-to-end supply for your laboratory workflow to ensure there are no concerns in your workflow and throughput. For COVID-19 testing, it has been critical to be vertically integrated. We are a single supplier and distributor for consumables, reagents, instrument software, and world-class service and support for laboratories running or looking to run COVID-19 testing. So when we think about partnering with Thermo Fisher, we really think about being a leading supply chain, world-class laboratory service and support, and providing that end-to-end COVID-19 workflow. Our assay is used globally as part of a CIVD kit, a US EUA combo kit, and as a component of many approved clinical laboratory workflows. I will show you how the assay is performing in our approved workflow, and later, Vijay Singh and Dr. Brooks will take us through how it is performing when at the center of other approved workflows. Overall, the TaqPath COVID-19 assay's design and chemistry consistently delivers highly specific and sensitive results. Applied Biosystems TaqPath COVID-19 kit delivers a high throughput, highly sensitive, and specific multiplex solution with expanding approvals for instruments, software, and streamlined sample prep. Clinical laboratories can now analyze patient specimens collected through 6 different methods, as you can see here on the slide. New sample prep protocol options deliver a 20% reduction in plastics by reducing the number of wash steps and the sample input from 400 microliters to 200 microliters, allowing double the amount of purified RNA. The Applied Biosystems TaqPath COVID-19 Combo Kit is now approved for use on all 7,500 systems, the QuantStudio 5 96-well 0.1 and 0.2 milliliter and 384 well blocks, and the QuantStudio 7 384 well block. And then lastly, on analysis and report, the new Applied Biosystems COVID-19 interpretive software helps your lab decrease analysis and interpretation time and eliminate subjective interpretation by automatically interpreting the data results from the combo kit, reducing risk of user interpretation error. You'll see here as we talk about the 6 approved sample inputs, one of the biggest bottlenecks in testing people for the novel coronavirus is around the shortage of swabs to take samples. The FDA announced that it would allow a broader range of swabs to be used in tests, including some made of polyester that should be far easier to manufacture, and these have subsequently been approved for manufacturers who have data that has deemed sufficient for extended approvals. We now have 6 different methods that can be used for our kit. The main difference for this is really how far the swab is inserted, as you can see in the diagram. Next, we'll go into specifically how the assay was designed. The combo assay includes 3 assays targeting regions of the SARS-CoV-2 regions with low genetic mutation risk, ORF1ab, and the S and N protein genes that have ongoing high sensitivity. The assay was intentionally designed specific to SARS-CoV-2 genome that are not present in other coronaviruses such as SARS, MERS, and bat SARS-like coronavirus, meaning our assay will retain higher specificity than assays that target other mutations. A clinical evaluation was performed to evaluate the performance of TaqPath COVID-19 kit using nasopharyngeal swab and bronchoalveolar lavage specimens. A total of 60 contrived positive specimens were tested, 30 positive NP and 30 positive ball specimens. In addition to the contrived positive specimens, 60 negative specimens were tested. In our studies, we have observed no measurable impact on sensitivity or specificity in the presence of common interfering substances, such as nasal administered drugs, blood, H1N1, and other allergy types of medications. Here we're showing the LOD concordance across all 96 well instruments. Nasopharyngeal swab specimens were spiked into purified full-length SARS-CoV-2 viral RNA at 3x and 1x LOD level. A LOD of 10 GCE per reaction, or genomic copy equivalent, for the combo kit was achieved using all 96 well instruments. In addition, an LOD of 10 GCE per reaction was achieved using both QuantStudio 5 and QuantStudio 7 384 blocks. Lastly, I want to show you in terms of our performance data was a FIND study that was recently released. FIND stands for Foundation for Innovation New Diagnostics. It's a global nonprofit organization that helps deliver diagnostics to combat major diseases affecting the world's poorest population and is a partner institution to the WHO. FIND performed an independent evaluation of commercial kit performance conducted at University Hospitals of Geneva. The primary objective was to verify claims analytical sensitivity, or LOD, using contrived specimens, and the secondary objective was to determine diagnostic accuracy using previously characterized clinical respiratory samples. They used 50 positive and 100 negative. The TaqPath COVID-19 CIVD RT-PCR kit achieved excellent results of LOD 1 to 10 copies per reaction with 100% sensitivity and 100% specificity. We thank FIND for including us into this study. Next, I'll go into the workflow. It's my last slide just to cover some of the questions that we've been receiving in terms of throughput. On every 96 well plate, you can analyze 94 specimens for COVID-19 in under 3 hours. On every 384 well plate, either on the QuantStudio 5 or the QuantStudio 7 384 well block, you can analyze 382 specimens for COVID-19 in under 6.5 hours. We see that one Kingfisher Flex instrument can feed either two 96 well block or one 384 well block instrument, as seen on this slide. With two full-time employees working simultaneously, you can achieve a workflow to analyze 2,820 specimens in one 24-hour shift. For laboratories who can scale with technicians in laboratory space with four Kingfisher Flex instruments and two 384 well instruments simultaneously, the workflow can even go up to over 12,000 specimens in a 24-hour shift. To help minimize instrument downtime during this challenging time, regardless of contract status, we are expanding access to our augmented reality and desktop remote support tools to all Applied Biosystems, Invitrogen, and Ion Torrent instrument customers. We've heard the outcry for the need for a high throughput solution to assist to get back to work programs and reopening of school campuses. We've also seen the demand of testing increase as infrastructure has scaled and reimbursement has increased. In response, we are developing a COVID-19 high throughput solution to provide and allow for customers to scale with ease without the burden of considering how to track samples, write scripts and protocols, or integration into their LIMS. So no matter the workflow or situation our assay is put in, you'll see that it's consistently delivering high specific and sensitive results. So I just want to thank everyone for listening on the chat. And lastly, we're continuing to scale both our testing manufacturing and expanding our approved workflow to enable more laboratories to perform COVID-19 testing with new sample inputs like saliva and high throughput instruments to assist with back to work and back to school programs to increase capacity. Thank you for the time, and I'll pass the presentation on to Vijay Singh, who will speak to his development on the Quant Studio 12K Flex.
All right. Okay. Thank you, Lauren. And so yeah, I'll be talking today about our experience here at Healthtrack RX in very quickly developing and incorporating a COVID-19 diagnostic test using the TaqPath kit and incorporating it basically into our existing workflow while pursuing an emergency use authorization for the lab from the FDA. So Healthtrack RX, we are a clinical solutions company working primarily in the fields of infectious disease testing and toxicology, and we're located in Denton, Texas, right outside Dallas. Now, some of these next slides, they're sort of covered by Lauren, and we are inundated by all the news. So SARS-CoV-2, responsible for the COVID-19 disease or pandemic that the world is reeling under right now. It's a novel coronavirus, and if you look at the genomic makeup, it's very, very similar to the known coronaviruses, especially SARS and MERS. And I'm not going to go into the next few slides into the great details about the epidemiology of the disease and details of the pandemic and how it is affecting life in general. I'll start talking directly about how we developed a test. So what defines a good diagnostic test, or any diagnostic test, but especially a clinical one? So here are some of the features. One, the test should be relevant, right, to the patient condition, and it should be the results from that test. It should have a positive impact on any of the clinical decisions that are being made with respect to patient care, right? The test should be rapid, so you can have a great test with a very dismal turnaround time, and ultimately it would have no clinical relevance. And lastly, a good diagnostic test should be reliable, right? At all points, the data that is being generated, one should be able to put complete trust into that, and features like low false positives and low false negatives. And as we go through, I'll be talking about how the TaqPath COVID-19 assay or the kit fits all of these criteria and why we ultimately adapted it in our testing line. So it was somewhere towards the end of February, early March this year when we decided to start testing for COVID-19. And what we started with was one of the commercially available CDC tests. So the CDC test covers only the N gene of the SARS-CoV-2, and with N1 and N2, two different targets within the gene, and the N3 was rejected for various reasons. And what we immediately found was that the tests were not performing, consistently. With respect to N1 and N2, and we would see different results, even though both the targets reside within the same gene. And as we were performing these validations, within a week, the singleplex TaqPath assay covering the ORF1A, N, and S gene came out. So we naturally gravitated towards that because now we are talking about 3 separate targets within the genome. And so automatically that is a good test, highly specific for a single target. And here at Healthtrack, we have a very vast experience in using the high throughput nanofluidic platform, the open array on the 12K flex. But those are for our infectious disease testing, and those are really large panel tests. And we hadn't really performed any single pathogen, single target tests. But we were interested in that, and what we found was that the singleplex TaqPath assay was highly consistent in the results that we were getting. And as we were readying our EUA application to the FDA with that, Thermo Fisher came out with the multiplex TaqPath COVID-19 assay, and one can immediately see how that would impact the turnaround time and the high throughput capabilities. So we changed course again, and we decided to follow that for our EUA application. And ultimately, and I'll give you some details for that as we go forward. So it was April 24th that the FDA granted the EUA to our lab at Healthtrack. And since launching the test, we have performed successfully over 220,000 COVID-19 tests. So how is the EUA granted to us different? It's in the sense that, we have, it was technically a bridging study or a bridging experiment in that our extraction methodology and the testing platform, the machine that we were using, are different from what was approved by the CDC or what was performed by the CDC and approved by the FDA. So after sample collection, our total RNA extraction is on a Hamilton Nimbus or a Hamilton Star fully automated system. And the actual real-time PCR is being performed on the QuantStudio 12K Flex system equipped with a 96-well block. And that is for what the data was submitted and the EUA granted to us. So getting into the details of what I showed you, the 3 properties of a good diagnostic test, and how the TaqPath kit follows that for us, right? That the good diagnostic test is relevant. And right now, when almost all of the states have lifted up their lockdown measures, this test is more relevant than ever. And not just the recent reports in the news, what we have noticed is that there is a definite uptick in the number of positive cases. And this surge, as the schools should open towards the end of summer, people need to get back to work. There is an absolute need to, have the people and the students reintroduced back to their work, their school, economic activities, educational activities in a safe and a reliable manner. And so there is no more there is no more important time than this to have, a test for this virus. How is this test rapid? Now, we had tested both the singleplex and the multiplex versions of this kit. And what this gives you is an absolute ability, depending on the availability of the nucleic acid, to perform 94 individual tests every hour. And, compared to the CDC kit that is available out there through various commercial vendors, it allows you only 30 individual samples to be tested at any given point. And so the rapid nature, and again, I'll briefly touch upon the 384 well format also, and that just gives you 4 times the capability. So how was this test reliable? What we found was that the TaqPath kit or assay was highly sensitive. And in fact, you can see in the data here, we validated it right down to 10 raised to power 0.1 RNA copies per microliter. These tests were performed by spiking sputum samples with the positive control RNA from the SARS-CoV-2. And we decided to go with the sputum for this round was because sputum can be a particularly difficult matrix to work with. But what you see here is that we find very good linear correlation with a 100% detection range. And we also tested right down to 0.025 copies and with about 95% detection range. And our very first 5 positive, 5 negative samples, we had an interlab correlation to test for that. There was another lab that sent us those 5 positive and 5 negative samples, and we tested them out in a blind fashion. And again, 100% correlation. We were able to detect that the correct positive and the correct negative samples. And since then, we have done the same for at least 4 different laboratories out there. So we are highly confident in the data that is being generated and the results that we are seeing. At the same time, while we were validating the test, we also did a test for the specificity of the assay. And we looked at previously known positive samples for a number of respiratory, viral, and bacterial pathogens, and which basically contribute. Now, this is the list that is in front of you, is the data that was submitted to the FDA. We tested a few more pathogen positive samples later, and we found that the TaqPath kit or the assay does not cross-react with any of these known positive or respiratory pathogens. And that increased our confidence with the test. Another example of how sensitive the test is, while we were, developing strategies to have the COVID-19 testing, we were looking at different extraction strategies. And in one of that, what we did was that we started diluting the samples to look at the sensitivity of the test. And the results are in front of you. One of the positive samples, we started diluting the same 1:5, 1:10, right up to 1:50, and the results are in front of you. The TaqPath COVID-19 assay was extremely sensitive. So even up to the 1/50th dilution, we got great signal. And the next one is quite an interesting example of how sensitive this test is. So we had reported out a sample as positive, and about 2 or 3 days later, the doctor's office calls us saying that, the patient had actually submitted their sample a week before it came to us to another lab, and a big lab that had a very, very disheartening turnaround time of nearly 10 to 14 days. And so that's why they didn't have the results. They were not feeling very well, and that is why they had sent the samples to us. Their sample from the other lab came back as negative, right? And so they were basically contesting our results. And I really cannot explain these results. So what we did was we knew that they were using the N1, N2 assay from the CDC kit. And so what we did was that we performed the test in parallel using the commercially available CDC kit and the TaqPath COVID-19 kit on the sample at the same time on the same machine, keeping all the other conditions consistent. And what we find, you can see that the N1, N2, we even performed the N3 target, which is not recommended. And the sample actually tested negative, but with the TaqPath 19 assay, it tested positive. And we had it correlated with another lab, just so that we are sure that it's not a false positive, and came back positive from one of the other labs that we knew was using the Thermo Fisher assay. And so this is what we have seen, with a lot of other commercially available kits, and that was the reason why we have, consistently used this kit, was that the number of inconclusive results that we get with the other assays is very high, right? So if we were running the N1 and N2 assay and the result is inconsistent with respect to only the N1 target showing up, or the N2 not showing up, or it's the N2 and not the N1 showing up, and the result would be reported out as inconsistent, I would say in 100% of those instances, the TaqPath 19 kit gives us an absolutely consistent result where we can confidently call it either positive or negative. So with that, what are the things that we're working on right now is there is a massive influx of samples into the laboratory, into our laboratories right now. And what we are trying to do is incorporate automation at all of our levels of our workflow. And we are currently trying to validate the 384 well format for the QuantStudio 12K also. In the meantime, we are partnering with a lot of local research groups within the DFW area, and to test the asymptomatic population for both antibody serology testing and PCR testing in parallel. And a paper will be coming out soon with our collaboration with a group at University of North Texas, where they're looking at an experimental platform using paper spray mass spec. And in fact, they report a very high correlation for our positive and negative samples. So samples that were provided to them in a double-blind fashion, they're able to correlate them with their experimental methodology to 95% or higher. And another paper that we are coming out very soon is that we have been running a lot of our samples in parallel on our open array platform for the respiratory panels. And so we'll be coming out soon with our data on the co-infection rates, or the bacterial and viral co-infection rates of COVID-19. So to summarize, the Thermo Fisher COVID-19 TaqPath assay has all the hallmarks of a good diagnostic test, right? It is relevant in this pandemic, where the need now is to have people resume their economic and educational activities in an absolutely safe and reliable manner. The test is rapid, like I said, 94 individual samples with the multiplexing capabilities. And it is highly reliable. As I showed you the data for the high sensitivity and specificity of the assay. And like I said before, so the number in front of you was current as of, I would say, 10 days ago. We have very reliably and successfully tested the kit on almost 220,000 different samples since we launched the test on 16th of March. And with that, I would like to thank the whole infectious disease team here at Healthtrack RX, which has been working nonstop in providing all these results out. And thanks to all of you for listening. And I would hand it over to Professor Brooks for his presentation. Thank you.
Professor Brooks, if you're speaking, we can't hear you. You may be on mute.
I was. Thank you. All right. Thank you, Vijay. What I was saying was that Vijay and Lauren covered a lot of ground with respect to COVID-19. So what I'd like to do is kind of describe what we've done with respect to using the TaqPath assay in our EUA, which is really focused around adding another biomaterial that we think is an excellent source for testing for COVID. And this innovation under pressure quote actually was from the President of the United States when he had announced the EUA that we had received for the first saliva-based test. And to be honest, we can't take all of the credit here. In fact, we take the smallest piece, because without an assay like TaqPath COVID-19, without extraction chemistries that have been optimized, and without preservation agents and really collection-based technologies, the only way to address supply chain issues, what we focused on early in testing was getting patients to the test. Now we can get tests to the patients with home collection, is really where a lot of the innovation comes from. You know, we're applications people, we put it together, we built the process, and we put it in play. So we're not going to cover anything other than we all know a lot about SARS-CoV-2, which causes COVID-19. This is a molecular-based approach, not an immunological-based approach. We actually are working on immunological detection from saliva, not a topic for discussion today, but a concept where we can actually use one biological sample to answer both parts of the question. We know a lot about coronaviruses. We're learning a lot more about SARS-CoV-2. I'm not going to review a lot of this, but I think the one thing that I want to point out with this slide is that, you know, testing as a function of detection doesn't answer a lot of the questions that we need to know about the virus. And Vijay's point with respect to why testing now is even more important than a few months ago is that we've spent, you know, a lot of our time developing assays with 99% specificity and sensitivity on 2% of the population. Now we need to look at what's really required and looking at pooling approaches, looking at creating more rapid or more efficient pipelines or workflows, because we have to look at overall sensitivity in 90% of the population, which is a very different lab-based approach. Not to mention, we don't know what the level of viral load is responsible for infection, right, or making people infectious, especially in the asymptomatic population. So RUCDR is a next-generation central lab provider and diagnostic services provider. We're a leader in biobanking, and we're self-proclaimed biomaterial experts, and that's how we got to saliva. We have worked for 20-plus years in helping pharma, biotech, academia, foreign governments, U.S. government, figure out what are the best biomaterials to collect, how do you best preserve them, and how do you get the most out of them. So we were able to leverage a lot of data, not on COVID-19, but a lot of data on what saliva can be used for, how we optimize with certain chemistries, how we optimize that with qPCR to get at the application of what we use TaqPath for. The other interesting thing is RUCDR started as a grant and contract research organization. We still are. So our ability to get to samples as well as private sector and state and national level programs to generate data was something that was really critical. And one thing to keep in mind is that our concept of managing the sample life cycle is really critical here, because again, you look at just the assay in a bubble, there are lots of things you need to take into consideration: the efficiency of collection, the stability of that sample, the safety of that sample, then the process that Vijay and Lauren have talked about, and then the storage of that sample after the fact. There are different environments that actually could potentially lead to a change in that sample. So as we start to develop, all of these biobanks around COVID-19 to do additional test development and other biomaterials, we really need to keep this sample life cycle in mind. The way we look at these things: science first, process second, automation third. This is a standard process that we use in the lab. The difference here in our process is we're using chemogen chemistry. We've also looked at the Thermo Fisher chemistry for extraction, but this is what we had in the lab. And we were looking for an assay, and we already were working on solutions for collection and preservation. But I think the transition above this dotted line of manually processing samples, using things that you have in the lab, eyeballing for QC, doing some QC that's rather blunt, is not going to get us to that high throughput solution, which are more automated approaches, using tubes and plastics that are automation friendly, incorporating QC tools, so that we can ultimately then go to any biomaterial and be able to process it. And that's what we like about most bead-based extraction chemistries, is that we can adjust them, whether it's pH, whether it's lysis solution, to be amenable to all different kinds of biomaterials. And this is just a sampling of all the different kinds of biomaterials we work with on every day, everything from tears to tissue to FFPE, stool, the nasopharyngeal swab that you see here, and also with saliva. And one thing that we need to keep in mind is that what we're looking at now is really unparalleled, as everybody knows. Imagine the some 350 million people in the US in 2019. There were almost 130 million hospital visits, 900 million doctor's office visits, and the 13 billion diagnostic tests were performed. Less than 10% of those were molecular analyses. And if you look at the scale and the scope of people being tested multiple times, if we were to just collect 5 different biomaterials or test someone recurrently, that's 4.4 billion samples. And imagine if you do that now every year as we live with coronavirus over an average lifespan of 50 years, that's 220 billion samples. So we know that molecular samples will exceed the number of paraffin blocks just in tissue, surgeries, biopsies alone, but we need to incorporate that with also self-collected samples. So if you look at direct-to-consumer, and the reason I'm drawing this analogy here has to do with saliva, because a lot of these applications, especially from where we made the connection, are at home or self-collection to be able to do this. And these numbers of samples are growing in leaps and bounds. Now, everything is on hold. All of our lives are on hold because of COVID. But this is something that we're going to revisit. And as we look at the fall with COVID and influenza, this is something that we're going to have to look at from a testing perspective where all of those samples become relevant. Lab automation innovation is also really critical. This was a collaboration, an automation collaboration that we did that allows us with an instrument the size of a New York City one-bedroom apartment kitchen to do 10,000 extractions a day, hands off. And this is the kind of work which takes what Lauren was taking to the next level, and she talks about additional innovation for high throughput as we start to screen large populations. Certainly once we have a blessing from the FDA to do those kinds of things, this is the kind of innovation that's needed. And we're going to see samples collected across all different healthcare markets. So what we're seeing now are all of these different kinds of companies come up with COVID-based testing approaches, or at least for their community, incorporating COVID into the kinds of tests that they do. So at-home testing for COVID-19, until recently, there was genealogy, there was entertainment through different apps that you could have in different places. There's health or risk assessment through 23andMe and Ancestry's new healthcare platform. And now, sadly, we can add infectious disease to this. And even though there are immunological-based tests that people do for infectious disease like HIV from saliva and others, COVID-19 is going to become a regular part of that regimen. Now, what's critically important in biosample management and COVID testing are the three segments that are the same for any biomaterial sample collection. And this is my own personal opinion, is where false negatives come from. They don't come from the assay. Thermo does an outstanding job in design and silico. The quality that they have and the control, as well as many other manufacturers, is outstanding. We rarely get a product that doesn't meet that criteria. However, what they can't control for and what we're responsible for as the lab, in this instance as a clinical lab, is making sure that sample is collected properly, that it's processed and extracted properly, and that it's stored properly. Because if any one of these things is not done properly, and each has a different level of variance that allows you to still get a positive or a change, that's where we see when there are false negatives, even potentially false positives. This is where the error comes from. So what we were looking for to build a process was to make sure we had the most robust sample collection. So again, for saliva, for nasopharyngeal swabs, which I'll, the comparison here of nasopharyngeal versus saliva is for nasopharyngeal, you have a healthcare professional that's at risk, very close to your face in taking the sample. You use a lot of PPE, which is also really limiting. And you're also limited by the surface area of what's on the tip of that flock swab. So granted, where the nasopharyngeal sample is taken from, the virus loves to live. It's dark, it's warm, it's mucousy, outstanding. But if you don't touch that swab in the right place, or the surface area of that swab, depending on what you touch, is limited, you are limited, even though the sample arguably could be potentially more concentrated. When you look at collecting 2 mLs of saliva, and on the right-hand side is the Spectrum saliva collection device, and you spit to that line, 2 minutes, you spit up to that black line, you screw the cap on, the blue preservation agent drops down into the sample, and now that sample is stable for transport, safe. The virus is inactivated, as well as the RNA preserved, that you're going to more reproducibly be able to spit into a tube than somebody that's going and necessarily taking a swab, because also there are differences in patient, how the patient responds. Now, when we did this through the first drive-throughs, once we got our EUA approval, we went from a 7 a.m. day to a 4 p.m. collection day or 5 p.m. collection day to a 7 a.m. to 1 p.m. collection day, and did 4 times the number of samples and reduced PPE by 90%. And those stayed in the hospitals where they were belonged for taking care of patients. So the patient experience is more robust, and the collection and reproducibility of collection is more robust. We reduce the risk to medical personnel, we reduce the usage of PPE, and we create a robust mechanism. Now, when we talk about screening in our EUA, a user goes through a quiz-based approach, a screening tool. We work with a number of telehealth partners, with a number of state organizations, people that are doing, their testing. You have to qualify a patient to be tested. So these guidelines are constantly being developed. And then you have to have a scalable system to support any kind of broad testing. And by population testing, I don't mean population screening. So what we've done is we've created a process that has a fail-safe mechanism to ensure that the test is that the test is collected properly, but it's also the right person. So when you start to do home collection, as the FDA explained to us as we went through, in a collaborative manner in putting this process together in our EUA authorization rather, is that you want to make sure it's the right person, but also the test is done in a safe manner, that if it's not done as indicated, no one's going to get hurt. And then also the sample stability and safety. So sample stability, if you give it to a carrier, we had to do temperature simulations, shipping simulations, obviously, to be able to show that that sample doesn't change in the context of shipping. One of our telehealth partners has a very streamlined approach for getting the kit. You actually go on a Zoom call, and then you get your report. Well, I know this is small that you can't see it. And then the reporting to the state and the regulatory authorities is automated, which is also a critical parameter for all labs doing testing, because without the departments of health and the federal government seeing these results and understanding it, there's no way we're going to learn what we need to be able to control and assess this pandemic. And as Vijay was saying, with sample positives on the rise, and we're seeing alarmingly the same thing from very different regions of the U.S. where we're testing now than we've been dealing with primarily before, being a lab in New Jersey and supporting New York, New Jersey, and the Northeast, a very different approach. So what happens in the laboratory? I think Lauren covered that really well. You extract viral RNA. In this case, it's from saliva. It's about a 40-minute procedure. It's all automated in that the saliva samples are moved to the plates that are used for extraction, and then liquid handlers are used to create a 384-well format. We then use qPCR. We love the concept of 3 genes. We can't tell you how many times this has helped us identify samples that would be at the level of sensitivity for other assays and giving us the confirmation that a multiplex assay is reinforcing the result that we are looking at. And then we look at the number of cycles, whether it's for CT or not. Thermo has a very simple to follow algorithm that helps us determine that. We validated that across samples. So what did our EUA process look like? Ours also, like Vijay was describing, was not a unique EUA in that we didn't create a brand new assay, but we made significant changes to the process. For us, it was the chemistry by PerkinElmer combined with the Thermo Fisher assay, but that really wasn't as much of an issue following the letter of the law. That would have mandated we go for our EUA, but it was really the addition of saliva and using saliva as the primary biomaterial. We did a whole number of contrived samples that were created, and what we submitted to the EUA was that, but also a clinical study as well. So we looked at 60 subjects that all had independently verified clinical results. So we had sent their samples to another reference lab blinded to make sure that the results that we got with saliva and nasopharyngeal swabs was the same. So these were all symptomatic patients across 3 different, they really weren't hospitals, they were ambulatory care facilities. And what we did at the same exact time was have a patient spit into the Spectrum saliva collection device, cap it up themselves, shake it for 2 seconds, hand it back to the healthcare professional. Then that healthcare professional donned and gowned, and then took a nasopharyngeal swab or an oropharyngeal swab from that patient at the same time. Both of those samples were processed within 72 hours at the same lab, at our lab. And to compare mass to mass, because really it would be hard to look at the surface area of the swab versus the amount of saliva, we used the same volumes. So the NP swab was put in 3.5 mLs of UTM. The saliva is 2 mLs of saliva and 1.5 mLs of preservation agent. So we're starting with the overall same total volume. We took the same amount of volume, 300 microliters of the saliva solution, 300 microliters of UTM, extracted them in parallel, and used 5 microliters of the extraction product for qPCR. So it's actually really unfair. I didn't change this on the slide to call this the old standard. The swabs are a standard, and they're an excellent way to test. It's not about old versus new. It's about identifying a way to get tests to people and also increase the robustness of testing. So as you know from Lauren's description, there's a spike in control. And what I just want to show here, I'm looking at the N gene, the ORF1AB, and the S gene in positive samples, that these are 5 different patients that you could see, depending on their own infectious journey, may be at a lower viral load, maybe at a high viral load. And we've seen patients that are on ventilators that have a lower viral load than a patient that shows up that's complaining they've lost their sense of taste. So I think understanding the clinical correlation to viral load is something that we're still learning a lot about. But from a testing perspective, this shows you that you can run the whole gamut depending on the person that you're collecting from, and this is from a swab. So these are selected samples just to show you within a single set of collections how that variable can be. And this also demonstrates how clean this assay is. From across the room, you could call a positive versus a negative. Now, if we look at saliva, we saw the same thing. So here are the same 5 subjects. You can see that they all have a different, different ends of the spectrum based on lower viral load, and the negative control data is exactly the same. And now if you look at positive and negative saliva, positive swab, positive control, negative control, you can see here in this summary, again, with selected samples, the positive controls are not at that lower end of the CT values, and that's a good thing because you want that positive control to be representative of potentially what that assay, limit of sensitivity is. whereas here are two examples of samples that, are, a much higher viral load. And again, for the no template control and your negative controls, this all checks out. It's been very robust in the thermal assay. But the striking thing is that if you look at the controls and the actual samples, you would expect to see a difference in, your controls. But if you look at the same 5 subjects for swabs versus salivas, we see a distinct shift to the left of just the fact that we think we have more virus or a more robust collection technique for those samples to show that saliva, and again, you're comparing 2 different biomaterials, so comparing, the viral load from each, you might expect a different results, and we're using the thermal assay quantitatively now. I know that it's not approved by Thermo for quantitation. We're doing that in our own lab and not in a clinical setting, but in a clinical trial setting for looking at this, but to show the robustness of saliva collection and processing. So if you look at the data summary, very similar to what Vijay had shown you in our contrived samples, we had 100% correlation between positive and negative. In the clinical samples, we also had 100%. So this was for nasopharyngeal compared to saliva in the same patients. Now, this was in a very controlled environment and controlled setting, but we did see 100% agreement on swab and saliva results, and we also had a 100% positive agreement in 1 to 2x LOD, and in some instances could see very low copies of samples. Now, what I will tell you is that we've worked on some clinical trials with healthy populations, and what we found is that in a population that we have of 1,000 people in a clinical trial, where every 2 weeks we collect an NP and a saliva sample, that we see a 96% correlation between saliva and NP, and these are in asymptomatic patients. This is data we're working on publishing, and we're working on submitting to the FDA as well. And that 4% difference is almost an even split. Half of them, 1.7%, are detected in NP but missed in saliva, and 2.3% are detected in saliva but missed in NP. And I think you're going to see a lot more data in matched studies that, can support this to show that they're both high quality tests. Both tests have to pass that certain, 96, 97%, a sensitivity, if you will. And those, by the way, were all patients that had low viral load if you're looking at the overall CT value associated with that. These were not high viral load patients. And as we collect more people and learn more about asymptomatic detection, I think what we're going to find, and again, this is just my opinion, that tests that are qualified as, highly sensitive molecular tests are going to hold true for detecting if anybody has that virus in any of these biomaterials. So we have a number of EU FDA, these are not approvals, they're authorizations, that's my bad, for both swab and saliva collection. So we're using the TaqPath kit in saliva and all of the other biomaterials that were on Lauren's slide. And then we had an authorization in May for home collection of saliva, which is really expanded the capacity for testing for people that are sick and don't want to go out, that have high risk, where their doctors want them tested, that are just afraid to go to areas where they want to be collected. So it's become a huge advantage for getting to people to the test. We use a number of different swab types. The saliva collection device is the Spectrum device. That's what's been validated in the EUA. We're using an automated chemogen chemistry for extraction. Like I said, our experience with the thermochemistry is also outstanding. Again, because of our development and the high throughput nature of our lab, this is what we had in place. The Thermo Fisher TaqPath COVID-19 kit is critical for success. So detection on the end, we wouldn't be where we are. We started with 384 well plates in the Quant Studio, and that was part of our authorization, strictly because of the throughput. We've also gotten approval. We're doing testing in every state in the union, but there are certain states that require specific licenses and approval. So we've done that. We're testing across a number of different applications. Like I said, we're working on a saliva-based immunity test. These are just copies of our existing EUA. And that's something hopefully we'll be telling you more about in the future. I had one additional slide, but in the interest of time, it was kind of a forward-thinking slide about how, personal biobanking is going to affect COVID and all these other things. Happy to leave that as a part of the deck if Thermo is going to circulate these. And I'm going to stop there and just thank everyone for their attention.
And thank all of you, Lauren, Dr. Singh, Dr. Brooks, for your informative presentation and your important research. We now are going to start the live Q&A portion of the webinar. Audience members, if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's take a look at the questions already coming in from our live audience. And we have quite a few great questions. Can the saliva test be used to detect asymptomatic and parasymptomatic subjects?
So, again, this is my opinion on this, is that the test doesn't know if a person has a fever or if they've lost their sense of smell or their sense of taste. And what we believe and have faith in is the sensitivity of the assay that's authorized by our EUA to how many copies per ml. We also don't know a lot about the clinical correlation associated with this. So what the appropriate level or sensitivity is to say that someone who's asymptomatic is infectious, we don't know that, or at least I don't know that. And these are some of the things that are being worked through. But in a straight answer to your question, we have detected COVID-19 in people that are asymptomatic as a part of research clinical trials and other people that have been authorized or medically approved for a test because of close contact or exposure.
And thank you for that. Similarly, another question about the saliva. Concerning the use of saliva for COVID-19 testing, it was shown that saliva samples of some COVID-19 patients were either negative or low positive, while the Nasopharyngeal swabs of the same patients were positive. How are you planning to address this issue?
So I'd have to look at that data. Again, this goes back to the false negative concerns that everyone has about any test. We could say the same about patients that test positive for saliva and negative for nasopharyngeal swabs. Early on, people would present because they were not feeling well. They would get a negative result. They would come back 2 days later and say, my conditions are worse. A little bit like the example of what Vijay had said, they're collected and they're positive. Now, I don't doubt that they weren't positive 2 days prior. Were they shedding enough virus, irrespective of the collection technique that was at the level of sensitivity for detection of the assay? That we don't know. All I can tell you, and is the paired studies that we've done, we've seen a very high correlation between NP, OP, and saliva. And some of it also has to do, like I said, with the method of collection, the experience of the person collecting, the preservation agent that's used for saliva collection. So I don't doubt that whoever asked that question has seen data that says there's a lack of correlation, but there's a lot more to look at to see why that's the case and not just say that saliva is not as good as NP.
Thank you for that. Now, Dr. Fossey brought up pool testing or group testing being a possibility. Are you working on any form of this? And if not, what makes this method more effective?
Is that question for me, for Andy?
I think it's open to whichever one of you would like to answer it.
I mean, I can answer that. To a certain extent, we have looked at pooling strategies, and again, the dilution that we had done on, and the data for which I had shown, was a part of that pooling strategy also. And yes, the concern would always be for the really low positive sample. And again, a pooling strategy would only work for the numbers that we saw in a 10 by 10 grid was the test would not be that effective if we are looking at a positive, overall positive percentage of 10 or higher, even 11% positive. That's when you have to go ahead and test all the samples in that pool. So yes, if the disease positivity rate in the population is, in our experience, less than 11%, that's only when any pooling strategy would be effective.
I want to thank all three of you. I wish we had time for more questions today, but I just want to remind our audience members that any questions not answered today will be answered via email. To all of you, would you like to provide any closing remarks for our audience before we close?
I would just like to thank everyone for joining us. We had over 1,100 people register for the webinar all over the world. So whether it's with the Thermo Fisher assay that's approved through CIVD or EUA, or some of the exciting new assays that you're hearing from both Vijay as well as Dr. Brooks, we're very excited to be in partnership with you in fighting COVID-19. Thank you for your time.
And again, I want to thank all of you for your time today and for your important research. I'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I want to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. And LabRoots will alert you via email when the webinar is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Be safe. Bye-bye.